Thank you very much, Jessica. I don't think people can see me very well, but I will proceed. We had an internet outage here in our building, so I decided to transfer to a lecture hall. So my talk today is about everything I have researched. Some is proper research, some is data that I collected from producers' fields here in Alberta, because Humic products are available. There are several of them out there in the market. No one knows what to do with them and whether they should be combined with any or everything, as some of the producers are doing. So this talk, I'm going to mostly focus on some work that we've done in a project here. As I said, some data we've collected from the greenhouse, not exhaustively, but also some of my on-farm experiences and data that we collected and what we found, so that I can be able to, at the end, give you some conclusions, although this work is still ongoing. So as Jessica said, my topic is Humalite, to apply or not to apply. So I would start by providing some background. All of us know that over time, the amount of fertilizer that we are applying in our field is increasing. And in the last couple of years, the fertilizer prices have just gone through the roof. So several approaches are being encouraged, or strategies, some people call them best management practices, are being encouraged for producers to employ. One of them is the 4R that we know: your right rate, right place, right source, and right time. We believe that if people judiciously use these, we will produce crops. We continue to produce the number of crops that we have here on the prairie sustainably. However, there is the use of biostimulants. And for biostimulants, it's very little information is available worldwide. I found a group of researchers in Italy and also at UC Davis in the US who have been looking at biostimulants. Reading through their works, I still found that it's still hard for people to know what right rate to use in these products, what right place, what right sources should be used, and what time these products should be applied in the field. I'm going to cover that in the work that we did here in Alberta. So what about stimulants? These are products that are more closely related to fertilizing products rather than to plant protection products. Why I'm raising this is because, for example, in the European Union, lots of people are developing these biostimulants. As you know, the abundance of biostimulants in the prairies is not a unique thing. Everywhere in the world, people are trying to see whether they can use these biostimulants. Why people are kind of thinking that this is more aligned to fertilizer products is because they want to avoid all that paperwork that it takes to register a plant protection product. However, some of these products behave as plant protection products, which I'm going to discuss later, about all these registering of products and what's happening in the European Union as opposed to what's happening here in the prairies. Well, these products that are available now, they are mostly aimed at optimizing fertilizer efficiencies and reducing input, which is what we try to look at in our research. They can either be microorganisms or they can be plant-derived. When we talk about microorganisms, I think most of you have heard about the product Envita. And then there are a whole suite of humic substances. And then there are also some, especially I think Corteva has some that have C extracts or other extracts that are being applied in the field. One of my students came and asked me, what's the difference between a biostimulant and an amendment? I was looking through literature and I found this interesting definition. It says that at least the people working in the biostimulant world have agreed that any product that is applied and it doesn't exceed 100 liters or 100 kilograms per acre qualifies as a biostimulant, while the others that you can apply in that. But there is, for sure, there's some interaction, because I think people do carry small quantities of manure and apply in their field, and they do not think that is a biostimulant. So any product that people have that is in this sphere, this group of researchers, mainly out of the US, have agreed that those they would consider as biostimulant. I wanted to just put that out there because I think people are also asking, okay, then what is an amendment now? Okay. Biostimulants, based on these researchers, they aim for the following reasons. They aim either to increase nitrogen use efficiency in a crop, help crops tolerate abiotic stresses, increase crop quality traits, or in the presence of humic substances, they are believed to have some chelating properties. So people believe worldwide, I'm going to show you my data, they believe that these humic substances or biostimulants will kind of like chelate and hold nutrient in place around the root zone and make it available for the crop. I wanted to show you this cloud diagram. It's from Professor Patrick Brown at the University of California. And they have produced a book that is out there. And they said, in summary, they believe that this is all the functions of biostimulants in plants, whether they are microbial or they are plant-derived, okay? I just wanted to put that out and put a link to this. If you are interested in reading more about biostimulants, you can check this book. It has paragraphs. It has different aspects that cannot be covered in a single talk. Okay. What they naturally believe, the people who work in biostimulant world, they believe that to develop a biostimulant, it starts with an idea, as shown here in this diagram. So they believe that if you start with the idea, then you start to develop that product, and you do several trials, and then you have like a feedback to the people who are involved in the development process. And then it goes through a quality control aspect, where they think whether this thing would cause cancer, is it healthy for people, animal, the environment, and then you proceed to a field trial. And throughout all of these, and then it goes through a regulation, and then there is an industrial or commercial production of these biostimulants. However, in the European Union, this is what happens. But as I said, in the Paris, nobody is actually, most of the products that are developed, it just ends with the industry, and the industry kind of try to find out what the mode of action is, and then they develop kind of a flyer or something to sell the product. And then most of the time, the product just goes directly, it's registered and goes directly into the field. I'd be proud to taking up my position here at the University of Alberta. I used to work at the University of Hohenheim in the south of Germany. And my job was to test out all these biostimulant products out in the field or in the greenhouse, and also to act as a third party. And then my report went to a regulatory office in Germany, where they kind of looked at my result and compared it against what the industry was showing. And then that's when the product was then put in the market or thrown out of business. So here in the Paris, we know that there's no standardized method to check even whether the biologically active compounds that people claim are in these products are actually in there. No regulatory outlines, and it is impossible based on what I'm going to share with humic substances, because the source of the humic substance really determines the activity of that substance. If a producer uses one product and it works, you cannot automatically recommend that product for another producer, which is very hard, because then how do you generalize results from one study to another? This is some of the shortcomings that I wanted to put out there, and see whether, now that we have government agency, whether something can be done, because I know several producers that are struggling with this product. One uses it 1 year, it works, the other one uses it the next farm or the next year, it doesn't work, no regulation. Okay. I'm just going to jump right into Humalite, which is the product that we have worked on. What is Humalite? Humalite is a naturally occurring kind of coal deposit. So thousands of years ago, when processes were formed and biological material was buried in the Peri provinces in Alberta, when coal was formed in that process, some of that material did not get transformed into coal. It got transformed into Humalite. And this Humalite that we are working with in Alberta has up to 70% humic acid content, which is really high compared to Humalite that you find in other places in the world where they are approximating about 50%. This Humalite also contains fluvic acid and some humin in it. On my left-hand side is a picture of how the Humalite looks like. It looks exactly like coal. And as I said, it can come in two forms, these Humalite or humic acids in Humalite. They can either be high molecular weight or they can be low molecular weight. And they have several side chains, which can either be a carboxylic, a phenolic, an aromatic, or an aliphatic side chain. The number and components of a humic substance, this number of side chains that I just enumerated, and whether they are high molecular weight or low molecular weight, has a lot to do with how active this source of Humalite would be, which may be explaining why we have all these haphazard results where some producers say the humic product they use work, others say no, it doesn't work. So we ran a project here for the last 3 seasons in Alberta, and our objectives were, one, to find out whether Humalite, if we added Humalite, we should be reducing our urea rate in wheat and in canola. And then we wanted also, from the point of business from the mine, to find out what is the optimum Humalite application rate for wheat and for canola in the field trials, because producers are going to the mine in southern Alberta, it's near Hannah, and what they're doing is they're just collecting buckets of Humalite and they're dumping them in their field. Nobody has taken the time to check whether do all the crops need the same Humalite? Should we be applying Humalite in some crops or not? And then the third objective was to find out whether there were some effects of Humalite on soil health indicators. In this presentation, I'm going to just look at soil pH. We did some incubation experiment, and I'm going to show you some of the results or some of the trends that we have seen. Okay. So for our field trial, we did a wheat, canola wheat in 2021, 2022, and 2023. We had 3 sites. There's an Abe research station in Alberta here. And then we had the Battle River Research Group site, which is a brown soil zone near Forestburg, and then the Gateway Research Organization site, which was the gray soil zone near Westlock. The experimental design was a split plot design. The main plots were sections with 0 Humalite, have the recommended rate of N, and then full recommended rate of N. And then the subplots were Humalite at different rate. We had 0 Humalite, 100 pounds per acre, 200 pounds per acre, 400 pounds per acre, and 800 pounds per acre. We collected agronomic data from these and soil health parameters. As I said, I'm not going to focus on that today. I'm going to mostly focus on yield and some economics. So when we did an ANOVA table, what we found was that mostly sites was very significant, and therefore I'm going to present the result in site. And then, of course, Humalite in alone in 2021 and 2022 did not show any significant differences, but we found some effect of Humalite in 2023. We didn't find any strong response of this Humalite on the crop proteins, and we're still going to look at in canola whether the fatty acid profile changed. So at the St. Albert site, in 2021 and 2022, here as what we found, the dark black bars is where we had our recommended urea. The light gray bar is half application of urea, and the white is where there was no urea. And that I'm going to try to stick to that color throughout the presentation. Okay. So in the wet year, we found that which is in 2021, we found that you find that where we had the recommended rate of fertilizer, we had yield that were higher, except in this case where they have urea had highest yield. And then when you look at a wet year, which was kind of almost a regular year, like 2022, you see that half urea kind of did really well. I'm kind of ignoring where we applied no urea because I don't know any producer that wants to grow their wheat without applying any fertilizers. So it looks like half the urea application rate in a regular year was best for wheat. But in a dry year, we found that yields did not really significantly differ from the control plot where we did not add any urea, and everyone knows, who is an agronomist, knows that in 2021, because we had very limited moisture, most of the urea that we applied in the field did not get broken down. So maybe that's why we didn't really see a strong impact there. In 2023, at the St. Albert side, here again, we start to see that in 2023, we had some drought at the beginning of the season, and then later on in the season, we had some rain come in there in July. And you can see that they have urea kind of outperform the full urea at 50, 100 pounds per acre Humalite application rate and 400 pounds per application rate, except for the 200 pounds per acre, where we need to still check this data for correctness, but you can see that clearly where we had fully fertilized plot, we had really high yields in this. For canola, you can see that for all the treatments, we can see that the full urea kind of had really high, had very high yields. Here is 2021 site again, BRRG versus GROW. BRRG is the brown soil zone, GROW is the gray soil zone. Here, below 200 pounds per acre, you can see that half urea and Humalite at 100 and 200 pounds per acre resulted in very high yields. And then when you increased the amount of Humalite that was applied in the field, then you started to see that you needed to apply fully. That means you have to apply the full urea application rate. In GROW, it was very straight out, although there was a lot of variability. This is field data. You can find that in GROW, at the GROW side, the half urea application rate had higher yields all through. In 2022, which was a kind of regular year, in the gray soil zone, here you find that we do not know what happened with these two plots, which had no urea in them. But if you look at where we increased the urea rate, you see that at 200 pounds per acre, it looks like you had to, in both cases, whether it's a wet year or a dry year, you had to fully fertilize to have increased yield. And then if you increase the amount of Humalite, then the half application of urea had highest yield. For the GROW side, we did not see anything really sticking out, except for at 200 rate, we had to fully fertilize. And then if you increase the Humalite rate to 400, then you had to reduce the amount of fertilizer that you were applying in the field. In 2023, which was last season, what we did in GROW and in BRRG is that instead of broadcasting the Humalite, we did sideband the urea. And then when we sideband the urea, we discovered that for the BRRG side, when you sideband, then you needed to put full fertilizer in it to have increased yields, as shown by the stars. And then for the GROW side again, even when you sideband, you see that the half urea had increased yield. Based on this information, we summarized there that for the black soil zone, which was in St. Albert, full urea plus 200 or 400 pounds per acre Humalite should be applied. In wet years, then you should use half the urea instead of the full urea. In the brown soil zone, we found the highest yields in canola when we had full urea plus 200 pounds per hectare Humalite and half urea plus 400 pounds per hectare Humalite. For yield, it was half urea plus 200 pounds per hectare and Humalite in a dry year. The story was not very clear for a wet year. For the gray soil zone, highest yields in 21 and 22, 2022 was half urea plus 400 pounds per acre. And then in 2023 was half urea plus 100 pounds per acre. Humalite. Based on this, the recommendations that I gave are strictly based on yield. I wanted to show you that we went further and did some economic analysis just to see what happens, especially for the growth site, where the results were kind of like very consistent across years. And so we did a net revenue generation. What we considered in this net revenue generation is we try to factor in only the yield, the cost of the yield, the cost of the urea, and the cost of fertilizer. The rest of the farm inputs we did not consider in this calculation. And what we found is that in 2021, for the growth site only, which is the gray soil zone, you can see that when we did not apply any urea, we had the highest profitability. Considering that this might be information that might be important maybe to some organic farmers, then I thought that I should highlight that. The second bar that you can see in this is the gray bars. And you can see that this gray bars is the half fertilizer application of the recommended rate. It was also consistently higher than the full urea application rate in terms of cost, in terms of how it pencils out for the producers. In 2022, there again, you see that not applying any fertilizer gives you the highest profitability, but then no producer does that. And then you can see the gray bars also showing higher, like giving you more revenues compared to the black bars. Same thing was observed for this site in 2023. This, I believe, has implications for some of the farmers that want to practice regenerative agriculture. Most of them want to just reduce. I think regenerative ag farmers are on a spectrum. There are people who want to like just go no fertilizer, but I also know producers who want to like reduce the amount of fertilizer that they are applying in their field. And therefore, I think that this might be a pointer for them, like how much should you apply or should you not just apply at all to get out still some yields and some benefits, especially if the farmer really wants to go that direction. Consistently, less revenues were generated when full urea was applied. You can see the black bars at the bottom. We did a strip trial where we wanted to look at what this Humalite we don't do with pea, because this current growing season we are going to have pea in the field, and therefore we had a strip that had no Humalite, and then there was a strip at 200 pounds per acre, and then there was another strip at 400 pounds per acre. I have brought out some pictures for you to just look at the plant root architecture. That is no news. We know that if you apply Humalite in the field, it increases plant health generally, especially root biomass. We also found that in this picture on the side, we also found that it also increased the number of nodules. And here you can see that the nodules are not single, but they are kind of like in clusters. So these clusters were greater when you had 400 pounds per acre Humalite. This trial we are going to put out in the field at 3 sites this season. And then we are going to see whether in the absence of N, because I think everyone here is aware that you don't grow pea with N. We're just going to inoculate the pea and have these different rates of Humalite. We're going to see whether nitrogen use efficiency in the field changes in the presence of Humalite. I wanted to show you also a preliminary study that we have carried out in the greenhouse, where I wanted to find out, because I know that there are producers out in the field there that are not only using urea, but they're also using Super U. So my question is, when you use Super U as opposed to urea, and you add Humalite to it, would it change the yield? So here we did a complete randomized design experiment in the greenhouse with 7 treatments from a control, urea, Super U at recommended rate, and then we reduced the Super U to 70%, the recommended rate. That's what the arrow stands for. And then we added Humalite to this, reduced urea and reduced super U. And then here is some preliminary results. Here, if you look at these, then you have your seed per plant in terms of gram here. And again, I have put a box in the bottom here that you can read, and you can see from T1 all the way to T7. And what we found was that there was no significant differences between the amount of seeds that canola was producing, whether we had urea or we had super U. Now, let's look at the reductions and all the other combinations. When we reduced the amount of urea to 70%, which is T4, you can see that there was a reduction in the amount of seeds that were produced per plant, okay? When you look at super U that was reduced to 70%, there was no reduction. So we can safely reduce the amount of super U that is applied without influencing how much seeds these canola plants were producing. This, as I said, is preliminary result. I'm going to still repeat this result in the greenhouse, which is ongoing, so that I can come to firm conclusions. No yield penalty, as I said, when super U was reduced to 70% and we added urea. This means that there's bad news for urea. It means that we can just reduce the super U to 70% and grow the plant, and there's no need for the producer to buy urea and apply urea in this system. Okay. Now I'm going to pivot and go into an incubation experiment that we are carrying out. It's ongoing. Why this experiment was done was because I had a lot of questions from people and contrasting information. And so the question is, does Humic or Humalite increase or decrease soil pH? I've talked to producers who are located in high pH zones, and they told me they use Humalite to reduce their pH. I've talked to people in the gray soil zone, and they said, no, we are using Humalite to increase our soil pH. So I did talk to Dr. Jeff Shuno, Professor Shuno at the University of Saskatchewan, and he said, maybe you should run an incubation experiment. This incubation experiment result that I'm presenting to you here is just the 7 days incubation experiment. Is by no means saying that this is the final result, because we've let this experiment now run for 3 months. It's still to be terminated. So what we did is that we went to different sites in the province of Alberta, and then we collected soils from the black soil zone, from the gray soil zone, and from the brown soil zone. And these are the treatments. So we added, there was no Humalite, then we added Humalite, we added urea, super U, urea plus Humalite, super U plus Humalite. We wanted to find out, when you incubate this, does the pH now go up or go down? Because it's important to give this information to producers. So this is the black soil zone result, and here you can see that with the treatments, that means once you start with Humalite alone, and then you go to urea alone, super U alone, and then urea plus super U, it looks like the pH is kind of increasing. And it's very different from the farm the soil that we got from Trushaw, which is really south of us, compared to where we had in St. Albert. We are looking into all the soil characteristics of these soils that we collected. And of course, I can tell you off the bat that the soil organic matter at these two sites were different, which can be why one of the arrows is showing a straight trend and the other one is not quite a straight trend. These results will be presented later in full. In another presentation, of course. For the gray soil zone, this is what we found. This first green bar is the pH that we got. I forgot to tell you, is the pH that we got when we collected the soil from the field and we're using the same lab. So this is in the gray soil zone. Here you can see directly that the soil that we got from Breton, pH immediately increased. Here, the pH did not really change, but with the fertilizers, it increased. But when you look at Humalite alone, in this case, the Humalite, except in this case that you see a little increase in pH, here, the Humalite did not change the pH at all. For the brown soil zone, here are some of the observations. Here again, we try to see like if you have fertilizer plus Humalite, there is a slight increase in pH. But if you have Humalite alone, at least after 7 days, we did not see any trend. Youngstown, the same thing, no changes. As I said, this is an ongoing incubation experiment. At the end, we would look at the soil organic matter of these soils, and then we would see after 3 months whether the soil pH changes with these treatments, especially in the presence of Humalite or not. Okay, the next question that I'm going to pivot to is leaf-applied humid substances. Do they work? There's all kinds of combinations going on there with humid products. I've seen producers mixing humic substances with their herbicides, with their fungicides. They're just mixing it with everything. And therefore, there's some work out of Poland that shows that leaf-applied humid substances do not increase yield. So what happened is that we had a group of producers out here in Camrose, and they had invited me to come over and check out their field. I'm just going to show you a couple of slides where what they get to me. I have not changed this or manipulated this. I'm just presenting it the way the producers gave it to me. So if you look at these slides, in the first column A, there are no humid products were added. B, they added 13 mils of fluvic acid per acre. So they sprayed it, and they have the data in May. And then, and mind you, this is winter wheat. This was a winter wheat field. So these were all like organized in strips. And then in C, what they did is that they added fluvic acid. They added this Castrac is a kind of Yara product that is supposed to increase your calcium in the field. Then they had 20 mils per acre of fluvic acid again that they applied on the 17th of June. And then they also had humic acids added. And then they did the same thing in the next strip, which is strip D. And what they added was 80 mil of Envita. So they had a double biostimulant in the field. And now, what they asked me to do was to come and measure. They said the plants look greener. We believe that they are photosynthesizing better. So what we did is we went out there to the field, to the producer's field, with the Series 3 photosynthetic meter, and we measured both photosynthesis parameters and gas exchange parameters. And amongst the gas exchange parameters that we measured, we mostly focus on FV over FM, which is the efficiency of photosystem 2. And then we did Phi2, which is how much light is absorbed by this plant and kept in the plant, not reflected into the environment. We also measured evapotranspiration and water use efficiency. I wanted to show you what we found. So remember, A is where we had nothing. And then B is where we had a fluvic product, C, fluvic and humic products, D, where we had those products plus Envita, and E is just the rest of the field. If you look at these, you see that photosystem 2 is not working better in the presence of this product because they all look the same. Secondly, if you look at Phi2, you see that the plants that were, oh my goodness, it looks like my letters are messed up. The plants that had these fluvic products were not harvesting more light, although the producers thought that it was greener. If they were not harvesting more light, you can see that all these bees, sorry, they should be here. There was no significant differences, okay? Here again, when we did evapotranspiration, we found that there was no significant differences between these. Maybe these A that they applied no product, less evaporation, the rest had humid products in them. Similar, even when Envita was present, and then E was the rest of the field. And if you look at water use efficiency, same thing, we did not see anything here. The A is where they applied no product. So you can see that no significant changes were happening when these products were leaf applied. Now I wanted to show this slide of the yield. Did yield increase? Because I think nobody really cares a lot about whether photosynthesis is happening there. And then you could also argue that we went at a point in time, so we might not have captured it. So what happened is, again, here, A is where we had no application of these products, and you can see fluvic acid, and humic acid with Envita. Here there was a little bit increase of yield, but if you factor in the cost of Envita, humic acid, fulvic acid, it's not really justified. So my advice would be that they found it in Italy, in Poland, that leaf-applied humic products do not really increase yields in crops. And we went and measured these out in the field. We didn't see any change in gas parameters. We didn't see any change in photosynthesis. And here is the yield from the producers combined. And we do not see any differences in yield. As I said, it's in kilogram per strip. So my general conclusion is that when we apply a field application of humic acid, which is mostly soil applied, we put it down with the seed, 200 and 400 pounds per acres have been shown in wheat and in canola, more so in wheat. Soil organic matter, especially the soil type, matters. Some soils, such as the black soils, I don't know whether it's really recommended. I mean, I want to get to the end of the project because we do not really see that benefit based on the economic analysis that I have not shown. The benefit of adding humic product or humic acids to black soils. Moisture and the crops does matter. Leaf-applied humic products are not effective. They might change how you might look at it and it looks greener. It doesn't lead to yield, so it doesn't pencil out. Humic products do differ, and the decision on whether to use them or not to use them should be based on economics. I want to acknowledge the contribution of my master's student who put together the slides, my postdocs who have helped with the data analysis, and all the funders, ARUDA, WestNet, Western Grains Research Foundation, and all the applied research centers who have contributed to the project. Thank you very much for your attention. I'm open for questions.
Thank you very much, Linda. We do have some questions. That was a very informative presentation. I believe there are about 6 questions, so I will start with the first one here. And the first question is, what is the influence of biostimulants on native microbiome?
I can only tell you that we did extract data from the root zone, and we did analyze it, and we did find that it wasn't really changing. At least that was from the black soil zone. We didn't see a shift in the microbial community, which is not surprising, because some of these humic products, they really operate where you have very low organic matter. That's where they really shine. So based on our Paris-specific result if you read in literature, it tells you that it changes the microbial community, which is from other parts of the world. But what I'm reporting is Paris-specific result. We didn't see any change for the black soil zone. We need to still assess the brown and the gray soil zone to see whether we see any change in microbial community. But for the black soil zone, we didn't.
Okay, thank you very much. The second question, first they say, I greatly appreciate the presentation on Humalites. It looks very interesting. Could you provide insights into why the application of Humalites showed no effects in the 2021 and 2022? Some effectiveness was observed in 2023, but subsequent years mirrored previous ones.
Yes, I can explain that. We had, I don't know whether that was captured in the way I explained it, but we are applying the same amount of Humalite in the same spot. So I believe that we started to see some responses in 2023 because of that consistent application. So in 2021, we put, for example, 20 to 200 pounds per acre in plot A. And then in 2022, I believe that there might be some additive effect of that repeated application in that spot. When we do our final analysis for this project, I would try to calculate not only a one-time application, but try to calculate that repetitive application, how much it would cost the producer and whether it is worthwhile. So that's an aspect that we have not looked into.
Okay, thank you. Another question here. For the field studies, what soil tests, oh, sorry, I just jumped down to the bottom here. I have to find that question again. For the field studies, what soil tests did you do before doing the experiment?
We did the normal fertility test where you do how much N, P, K, whatever, all of that on the list. Standard field test, and then you find out exactly how much N is in a plot. So we did like several soil cores, random soil cores across the plot, and then we put that together in the different soil horizons. We mixed them, and then we found out exactly how much N is in that field, and then we got a recommendation for wheat, and then we divided. So when we say half, the recommended N is half of that amount for that site.
Okay, excellent. With the Humalite experiments, was the nitrogen content of the soil of all plots determined before the treatments were applied? Just wondering about yields with zero fertilizer.
We did, as I said, through the plot. We did per block, not through every single plot. We have taken data at flowering for soil parameters that I have not shown here. But we do have every plot N and stuff like that, but this was too long for me to put it in here. We're going to try to analyze that and publish it, where we did the N at the beginning of the trial for the block to have a baseline, and then we did it again at flowering per plot. We did 4 spots per plot, and this has been pulled together, and we sent it to the lab. We just got that results back. So we're going to start to look at, so all I've talked about is the impact of Humalite on yield and not actually soil health, because that data needs to be analyzed. So although it doesn't produce yield, it doesn't increase yield, it might have some soil health benefit, but I can't speak to that now because we are still to analyze that data.
For sure. A further question here. For the Humalite applications on peas, are there plans to investigate herbicide field history to account for the possible impact of residual herbicides on root growth? There could be efficacy of Humalite on tying up the herbicide.
Ooh, I didn't think about that. That's a great idea. I don't think I have funding to do herbicide residue, but that's something that we could look into. The mine is doing a site experiment, and they keep saying that we should look at pulses more, that it might be a product for pulses. So that's why we are kind of taking part of the project this year from grow to the peas to just check whether those low soil organic matter fields would do better with the pea. But I think that's a great idea to look at Humalite and herbicide residue. It's something that is in the back of my mind. Thank you for bringing up that idea. I think it's something worth looking at.
There are a few more questions here yet. Lots of interest in your topic. Which soil pH levels were found to be better for biostimulant effectiveness, and how did soil physical and chemical properties affect the effectiveness of biostimulants?
I can speak to these only from what I have read. We know that Humalite, at least the product we are working with, is very acidic. So I am hoping that it would reduce soil pH for high pH soils. That is what I am expected to see, not increase soil pH. Humalite has some micronutrients in it and also has some carbon structure in it to it. So it's going to add some to the soil organic matter in some way. So I'm expecting that we should be able to see changes, especially like in the brown soil zones and also in the gray soil zones. I'm not expecting to see a lot of impact of Humalite in the black soil zone.
Okay. And are there any regulations or guidelines for the use of Humalite in agriculture? And how can farmers ensure they're using it effectively and responsibly?
Unfortunately, in the prairies, that was my shock flying in here from Stuttgart, Germany, I was shocked that products do not go through any regulation. As I said, with humic products, the source of the Humalite, the side chains of the Humalite really determines how active that humic product is going to be. And so if you have a manufacturer going to several sources and there's nobody controlling what the chemical active ingredient in this product is, it means that that product is sold every time and what is sold is different. I don't know all about the quality control here in the prairies, but producers are just overwhelmed with all the sources of Humalite. And as I said, that's why some years it works on some fields and it doesn't work in other fields. It's all these sources because there are several sources. I think North Dakota, South Dakota also has mines there that have humic substances in those mines that are also kind of extracted and sold on the market, and they have less humic acids in them, or they have extracted the humic acid in them. It's chaos in the market because every time we take, we know that what we are using in our project is from the mine, and we do a soil test on every batch, and we've seen that it's kind of almost consistent. So I can speak to this product, but the fact that I said, oh, if you apply 20 pounds per acre of Humalite from Hana, does not mean that you should run to the market now and translate that into a humic product that you can find in the next dealership. That is not true. I think that the government has to do something at some point about the humic product and generally most of the products that are out there. There's no control, there's no way our farmers are overwhelmed.
Mm-hmm. Yeah, there's a few questions here actually in regards to cost, wondering what the cost of Humalite would be per ton, or the increase of the cost increase of applying 200 to 400 pounds per acre.
The cost of Humalite at the moment is $300 per ton.
Okay.
So you need to do that in your, there are producers that are like, if it doesn't hurt, they don't mind, they're gonna still apply it. And they're like, if it improves my soil and I don't get a yield, but I can consistently get yield out of this field, that's fine for me. So it depends on your goals and your objectives. And as I said, please either broadcast it or put it down with your end. Do not side burn it for it to go close to the seed, it does burn. That's what we learned from side burning Humalite. If it gets close to the seed, it will burn your seed just like urea would.
Okay. Does Humalite have any effect or interaction with pesticides and residual effect?
I have no clue. I have not read anything on that at the moment. I don't really know anyone who is working on that. Everybody working on Humalite or humic substances are working on either reduce and input into the system or abiotic stress mitigation. I've not read any article that directly addresses herbicide or fungicide residues with Humalite. I have not.
Okay, thank you. Do you think that there would be do you think some of the benefits of humic acid will show up slowly, maybe in 5 to 6 years?
That is what we are hoping. We started in 2021. The part of the project that had the repeated application of the Humalite in one spot ended this last season. The mine has said we should hold on to the land. The thing is, it's very hard to get funding to do all the things we want to do on Humalite. So I don't know whether we'll be able to get money to do this long term and have some solutions, some answers for long-term studies. But we'll see. We've done only 3 seasons, and 1 year of that 3 seasons was a drought year. Mm-hmm. So I don't know. At the moment, I don't know.
Okay. Is crop stage sorry, I'm just reading the question. Is crop stage affects the performance of foliage biostimulants?
I can tell you that most of the biostimulants out of the research that I read from Europe, they applied it at fungicide timing when the plants were like flowering, and that was weed. They found no effect. Our producer here that I showed you, Fred went out there and Fred did 2 times in this winter weed in May and again in June. So he applied fluvic acid, humic acid, they were all leaf applied. As I said, it was quite a show. There was the drone guy who applied it. There was the person sending them the product, bunch of producers out there, and we went out there live and measured these photosynthetic parameters, two of which I have shown you. And we didn't see any significant difference in strips that had foliar applied Humalite in it, whether they had foliar applied Humalite or humic acid or only humic acid or humic acid and fluvic acid plus Envita. We didn't see any change in how the plants were photosynthesizing in these strips. And we didn't measure one spot in the strip. We measured at least 10 spots in the strips, and we brought it live and we sat there and had a conversation. And then when he did the harvesting, you saw how the yields turned out. So do it only if you think that it's profitable, but based on the research out of Europe, and this is a producer's farm, I didn't tell him like what herbicides to put in this system and everything. He just did it and applied it, and then we showed up and measured. And we found that there was no significance in photosynthesis or gas exchange parameters. The plants were all behaving the same. And then you see that the yield didn't, I mean, there was some slight increase in yield where you had Envita, but he told me it doesn't pencil out for the cost of the Envita, the cost of the fluvic acid and the humic acids that were applied. He told me it didn't pencil out. So I can only report to you what I know at the moment.
Yeah, thank you. And a further question here. Where the crop seems greener due to the addition of fulvic acid, did you measure the amount of nitrogen in the plants?
My student just collected those green plants, okay, and they are in the lab at the moment. We are hoping that we'll see something. I don't have that result to share at the moment.
Okay. What are your thoughts on humic products with amino acids, lysine, threonine? If having those amino acids added and promoted to speed up humic availability in the soil?
I don't know whether we need more humic products. The way humic products work is that they chelate. It means that they grab onto the nutrient and hold it in the vicinity of the root zone. This can go two ways. The nutrient can be chelated and made available to the plant, or the nutrient can be chelated and stopped. The plant will not be able to access this nutrient, which means the plant will be dying in the midst of plenty. Not with humic product per se, but some of the products that I work with in Germany, that's what they were doing. They would just grab onto the nutrients too fast. That means chelate it really hard from bonds that were too strong that the plants were unable to take up the nutrient. So I would say that maybe if you have humic products that have some amino acids in them, it warrants that some research be done before you go. I mean, every time something chelates something doesn't mean that it's a good thing. At least that's what we found in Germany from our work in Germany.
Are there any plans to research the effects of perennial forage stands and not just on annual crops, as Humalite products are really being pushed in turf management?
It would make sense for forages, just the same as it makes sense for greenhouse gases, because there's abundance of research out there that shows that Humalite promotes root growth, promotes plant height, promotes more biomass for plants, especially in the greenhouse. We've seen that. Root architecture, I mean, I could show you. I just felt that this was a group of agrologists who are dealing with producers. That's why I kind of skewed the talk towards what we found on farm. But we have data, me and my colleague, Dr. Malinda Tirakarana, he's doing all the greenhouse part of it. And when you have nothing but sun, some fertilizer and Humalite, we have the yield per plant going up. Like you get more plant and more yields, especially in wheat, you get more yields per plant. But then if you put it in the field that is a more complex environment, you get the root growth, you get healthier plants, you get more yields probably in a dry year, but you do not get that in our regular growing seasons. It's a more complex story.
Yeah, absolutely. Here's a comment. Someone says, I think it would be interesting to analyze potential longer term over the years changes in the soil bulk density and water infiltration rate to see if it if continuous applications of soil applied Humalite can make improvements to soil structure, which can help improve yields. And
yes, we took infiltration rates. As I said, this talk was more the above ground. We took data on water infiltration rate, bulk density, you name it. The list is 20, electrical conductivity of the soil, I mean, and so on and so forth, and nitrates, whatever. We have that, as I said, the data just came back from the lab. We're putting it together. We're going to analyze it and see, at least for this year, did it change from 2021? That's what we'll be looking at.
Right. Yes. So there is a residual effect of Humalite year over year. Is that what you were saying?
I was just saying that we are going to try to look at the data and analyze it in that light. Like where the water infiltration in 2021, did it change in 2023 after consistently applying this set amount or fixed amount of Humalite in these plots? Those are some of the questions that we are going to answer, but I don't have that data now with me to share with you. I think the students are trying to work on it and the postdocs. Yeah, I mean, I will be open to working in turf grass if somebody will give me money. As I said, it's very hard to find funding to work on something like an amendment. We got funded by ARDA, and we did put in a proposal to do some more microbiome work in the brown and gray soil. It got turned down. So somebody gives me money, I will look at it. We have the soil sample sitting in the lab. So if somebody gives me money, I will look into that for sure.
Definitely room for more research. Final question here. Is there current research being done or to look at measurable effects of Humalites on nutrient absorption, as well as in cereals and oil seeds crops?
My colleagues there in the lab, they have been looking at using NPK and stuff like that, and looking at the root architecture and stuff like that. That's all that I know that is being done. We have also added a colleague who is a proteomics person to look at how proteins are changing and what is actually happening. And at the moment, I mean, as per his last report, for Humalite in the field, he found that it looks like the plants are more efficiently using phosphates. It's just, I mean, it's new information. Don't hold me to it. I can probably send you his talk if you're somebody who is interested in proteins, which ones are upregulated. So what we did is we went out to the field and then we plucked up some leaves, several leaves per plot, and then he took that back and extracted DNA and proteins out of that, and he's looking at which genes are upregulated, which proteins are involved, and he said everything is pointing not to N, but it's pointing to P, and I was kind of shocked because I thought I'm going to be doing this and trying to see this end-use efficiency with Humalite, and now he's pointing us in another direction. So.
Okay. This is the final question, and then we'll let you go. What was the rationale for selecting the application rates for the Humalites in your study?
We started from zero. We didn't have no rationale. When we had this product, we looked at it and we said, let's look around and see whether anybody has tested this thing, and we found nothing. So we said we're going to just like incrementally increase it. Maybe we would find a rate that works. That's why we went for zero, and then we went 100, 200, 400, 800. I mean, no producer is going to apply 800 pounds per acre, but we said, let's put it there and just see what happens. Maybe there's a level of Humalite that is toxic for the plant, who knows? We started from zero, so there was no exact, we just increased it and said, let's see, maybe we hit it and miss it, or we see it somewhere in between, and then further research can be off of this now.
Excellent.
So that's how we came to that.
Wonderful. Thank you so much, Linda. Your talk has obviously brought a lot of interest, and I'm sure your contact information will be made available, is my guess, if people have further questions.
Yes, please reach out, and if you have more money and ideas, please come work with me or give me money and we can check out things. I don't mind. Thank you very much.
Wonderful. Thank you again. There will be